Okay, so good evening, guys. Welcome to the last and final session of the series of live sessions where we basically discuss questions from the course Fundamentals of Protein Chemistry taken by course instructor Professor Shagota from IIT Kharagpur. So what we do in these live sessions is that we discuss some of the old assignment questions, and we not only discuss the questions, we also discuss the concept behind those questions, that it's easier for us to solve similar type of other questions. And we try to keep these sessions as interactive as possible. So if you have any queries, any doubts, you can just unmute your mic and let me know, or you can type in your chat box if your mic is causing issues. And if I know the answer immediately, I will let you know. If I don't, then you'll have to give me some time. I'll get back to you as soon as possible. Okay. So with that introduction, let's start the first question for today. Which of the following sorry. So with the first question for today, which of the following is not a common application of fluorescence spectroscopy in protein studies? Number 1, measuring protein ligand interactions. Number 2, determining protein sequence. Number 3, studying protein folding. Number 4, detecting conformational changes. Choose the correct option. Option A, 1, 2 and 4. Option B, 2. Option C, 2 and 3. Option D, 4. I will wait for a few minutes. You can come up with the answer and the explanation. If not, then I will discuss and then we'll move on to the next question. Okay, so the correct option will be 2, which means determining protein sequence. So fluorescence spectroscopy is a powerful, highly sensitive technique used to study the structure, dynamics and interactions of proteins, but not their amino acid sequence. Okay. For example, let's analyze each option. Number 1 is measuring protein ligand interactions. So it's a very common application. Binding of a ligand to a protein often changes the local environment of aromatic residues such as tryptophan, tyrosine, unfin and alanine. And this leads to a measurable change in fluorescence intensity or emission wavelength, lambda max. So this is used to estimate binding constants, KD, and study binding kinetics. Going to statement 3, studying protein folding. That is also a common application. Protein folding, unfolding alters the exposure of aromatic residues, example tryptophan. So monitoring changes in fluorescence intensity or wavelength shift during denaturation or refolding gives insights into the folding pathways and stability. Statement 4, detecting conformational changes. That is also a common application because conformational shifts due to pH, ligand binding, temperature, altered fluorophore environments. It's for intrinsic tryptophan or extrinsic ANS Fred pairs or fluorescence is used to detect the changes. But statement 2, determining protein sequence, that is not a fluorescence application, because determining a protein's amino acid sequence requires sequencing methods like advent degradation or mass spectrometry, MS based sequencing. So fluorescence spectroscopy cannot directly reveal the order of amino acids. It provides only environmental or conformation information, not the sequence data. So if we talk about spectrophotometer or fluorescence spectrophotometers, basically they measure the fluorescence signature of an analyte in a sample based on its specific excitation and emission wavelengths. Now the fluorescence signature can be correlated to the concentration level of the analyte in the sample. A spectrophotometer can be used in basic and applied research for biofuels analysis, biotechnology applications, quality control, medical diagnostics, plasma monitoring, polymer analysis, all of this. Now principle wise, fluorescence is an emissions phenomenon where an energy transition from a higher to a lower state is accompanied by radiation. Now only molecules in their excited forms are able to emit fluorescence. Thus they have to be brought into a state of higher energy prior to the emission phenomenon. Now, molecule in its electronic and vibrational ground state can absorb photons matching the energy difference of its various discrete states. The required photon energy has to be higher than that required to reach the vibrational ground state of the first electronic excited state. The excess energy is absorbed as vibrational energy and quickly dissipated as heat by collision with solvent molecules. The molecule thus returns to the vibrational ground state. These relaxation processes are non-radiating transitions from one energetic state to another with lower energy and are called internal conversion or IC. From the lowest level of the first electronic excited state, the molecule returns to the ground state either by emitting light, fluorescence, or by a non-radiative transition. So upon radiative transition, the molecule can end up in any of the vibrational steps or states of the electronic ground state as per quantum mechanical rules. Now since radiative energy is lost in fluorescence as compared to the absorption, the fluorescence light is always at a longer wavelength than the exciting light, like Stokes shift. The emitted radiation appears as band spectrum because there are many closely related wavelength values dependent on the vibrational and rotational energy levels attained. So an associated phenomenon in this context is phosphorescence, which arises from a transition from a triplet state to an electronic singlet ground state. The molecule gets into the triplet state from an electronic excited singlet state by a process called intersystem crossing, or ISC. The transition from singlet to triplet is quantum mechanically not allowed, and this only happens with low probability in certain molecules where the electronic structure is favorable. Such molecules usually contain heavy atoms. So the rate constants for phosphorescence are much longer, and phosphorescence thus happens with a long delay and persists even when the exciting energy is no longer applied. So the physics behind fluorescence involves the different electronic and vibrational states that fluorophores can exist in. An electronic state is divided into multiple vibrational states, and photons that have energies in the ultraviolet to blue-green range of the spectrum can trigger an electronic transition from a lowest vibration in the ground state to one of the vibrational levels in a higher electronic excited state. As soon as the energy input from the photon, in other words the excitation, stops, the fluorophore molecule relaxes into the lowest vibrational level of the excited electronic state. The fluorophore remains in this state for some time, around 10 nanoseconds, known as the fluorescence lifetime, and then returns to the electronic ground state. This return to the ground state is associated with the release of energy, known as the fluorescence emission. Now the number of photons emitted by a fluorophore relative to the number of photons absorbed is called the quantum yield. A fluorophore with a large quantum yield like rhodamine will display a bright emission. The difference between excitation and emission wavelengths is called Stokes shift. Stokes studies of fluorescent substances led to the formulation of Stokes' law, which states that the wavelength of fluorescent light is always greater than that of the exciting radiation. Now the process that happens between excitation and emission is illustrated using Jablonski diagram, named after the father of fluorescence spectroscopy, Alexander Jablonski. If you see the triplet excited state, an excited state in which the unpaired electron spins occur when the analyte returns from a higher energy state to a lower energy state with the opposite spin. Singlet excited state is the excited state in which all electron spins are paired. So when the analyte returns from a higher energy state to a lower energy state with the same spin. Now a molecule's quantum yield is influenced by external variables such as temperature, viscosity of solvent, pH. Increasing temperature generally decreases because more frequent collisions between the molecule and the solvent increases external conversion. Decreasing the solvent's viscosity decreases for similar reasons. For an analyte with acidic or basic functional groups, a change in pH may change the analyte structure and therefore its fluorescent properties. Intrinsic fluorophores with the natural molecules with the natural fluorescence such as chlorophyll and the aromatic amino acids. Extrinsic fluorophores are those that are added to a sample to provide fluorescence or to change the spectral properties of the sample. Examples of extrinsic fluorophores are fluorescein and rhodamine, but there are many more. So each fluorophore has its own characteristic properties such as fluorescence lifetime, intensity and position of the emission wavelength. Thus each fluorophore will yield a unique fluorescence spectrum. A fluorescence spectrum is a plot of the fluorescence intensity as a function of wavelength. So fluorescent intensity F is dependent on both intrinsic properties of the compound, fluorescence quantum yield, and on readily controlled experimental parameters including intensity of the absorbed light, molar absorption coefficient, path length of the cell, and concentration of the fluorophore in solution C. Now, how does the spectrophlorometer work? So, the essential components of spectrophlorometer are a light source, an excitation monochromator, a sample cell or cuvette, an emission monochromator, and a detector. Fluorometers quantify biological analytes as a function of fluorescence. So, this requires a sample to be bound to a specific fluorescent agent and loaded into the instrument in a cell or cuvette holder. Now, the light source sends out light at the excitation wavelength of an analyte in the sample. Before it reaches the sample, the light passes through the excitation monochromator, which transmits a wavelength specific to the excitation spectrum of the analyte while blocking other wavelengths. Now, the light from the excitation monochromator passes through the sample containing the sample cell or cuvette holder and excites the analyte or the fluorophore. Now, fluorophores absorb light of a distinct excitation wavelength and emit or fluoresce light of reduced energy, thus a longer wavelength. The emitted light passes through the emission monochromator positioned at a right angle to the excitation light, and the emission monochromator minimizes light scatter and screens the emission light before it reaches the detector. So, the detector measures the emitted light, displays the fluorescence value, and produces the fluorescence signal of the analyte. So, the fluorescence value is proportional to the concentration level of the analyte in the sample. So, if you can see, this is the excitation. Sample cuvette, 90-degree, then grating slit, photomultiplier, sample detector, and now, it has been used for the direct or indirect quantification and qualitative analysis for measuring fluorescent intensity. It is relatively inexpensive and sensitive. It's approximately 1000 times greater than absorption spectrophotometric methods. Now, what are the applications? In the biosciences, one of the most frequent applications of fluorescence spectroscopy is the high precision quantification of DNA and RNA using ETBR as an extrinsic fluorophore. Another modern application is SMRT, single molecule real-time DNA sequencing. Its ability to produce long-read single molecules with high accuracy. It is predicted to be central to the next genetic diagnostic evolution. Now, proteins possess three intrinsic fluorophores, tryptophan, tyrosine, and phenylalanine. Although the latter has a very low quantum yield and its contribution to protein fluorescence emission is thus negligible. The main application for intrinsic protein fluorescence aims at conformation monitoring. For industrial applications, it is used in several industrial settings such as a fast, non-invasive technique in the assessment of contamination. For example, it has been used to detect contaminating organic compounds in groundwater after hydraulic fracturing for gas exploration. In chemicals, an important chemical application of fluorescence spectroscopy can be found in the field of nanoparticle synthesis for potential medical uses such as drug delivery. For environmental monitoring, the technique also has wide application. High resolution fluorescence spectroscopy and 3D excitation emission metrics fluorescence spectroscopy are used to characterize dissolved organic matter in these samples and then based on that optimize treatment process for landfill. For pharmaceutical, it is also used in pharmaceutical field to analyze drugs, like the analysis of co-formulated tablets, prescribed as cholesterol medication. In agriculture, spectroscopic techniques are widely used in the identification of different crop varieties. Likewise, total luminescence spectroscopy can be used by tea manufacturers as a quick, affordable, and an objective alternative to employing trained tea testers to discriminate between similar types of tea. Now, if you talk about the green fluorescent protein, this is gene from jellyfish Aquaria victoria, right? So basically, it's an autocatalytic reaction of amino acids in the protein cyclization and oxidation by O2. And the rigid protein solvent enhances the fluorescence quantum. The final fluorophore contains a series of conjugated double bonds. Now, So here protein of interest can be fused with different fluorescence proteins, and it is used to basically localize proteins within the cells in vivo. Okay. For example, present on Golgi and plasma membrane, this protein which is labeled with YFP, calnukes which is labeled with sand fluorescent protein. Similarly, calmodulin cytoplasm labeled with CFP. Quantum yield and lifetime. We have already talked about that. Fluorescence lifetime basically a measure of how long the molecule remains in the excited state, and limits that influence the excited state. Intrinsic fluorescence of proteins. You have talked fluorescence spectrum of amino acids in water. Spectra of several proteins. This is mostly due to tryptophan. In most proteins, the fluorescence is dominated by tryptophan. For example, in BSL, bovine serum albumin, tyrosine and phenylalanine fluorescence is mostly quenched by interactions with surrounding protein residues. So if you go through these papers, you will find an example of the use of fluorescence spectroscopy in biology. So I will not go into too much detail for these papers. You can just go around in these papers and check it out. So you have talked about fluorimetry, which is basically a type of luminescence caused by photons exciting a molecule, raising it to an electron excited state. It's an optical phenomenon in which the molecular absorption of energy in the form of photons triggers the emission of fluorescent photons with a longer wavelength. Phosphorescence is a specific type of photoluminescence related to fluorescence. Unlike fluorescence, a phosphorescent material does not immediately re-emit the radiation it absorbs. The slower timescales of the re-emission are associated with forbidden energy state transitions in quantum mechanics. As these transitions occur very slowly in certain materials, absorbed radiation may be re-emitted at a lower intensity for up to several hours after the original excitation. So the mechanism of fluorescence, fluorochromes will only fluoresce if they are illuminated with light of the corresponding wavelength, and the wavelength depends on the absorption spectrum of the fluorophore. It has to be ensured that an appropriate quantity of energy is delivered to elevate the electrons to the excited state. After the electrons are excited, they can dwell in this high energy state for a very short time only. When the electrons relax to the ground state or another state with a lower energy level, energy is released as a photon. As some of the energy is lost during this process, light with an increased wavelength and lower energy is emitted by the fluorochrome compared to the absorbed light. Okay. So spectroscopy provides two types of spectrum: excitation or absorption spectrum and emission spectrum. So understand the difference between fluorescence and phosphorescence requires the knowledge of electron spin and the differences between the singlet and the triplet states. So according to the Pauli exclusion principle, two electrons in an atom cannot have the same four quantum numbers. You know that principle: azimuthal, magnetic and spin quantum number. So only two electrons can occupy each orbital where they have the opposite spin states. These opposite spin states are called spin pairing. So because of the spin pairing, most molecules do not exhibit a magnetic field and a diamagnetic. In diamagnetic molecules, electrons are not attracted or repelled by the static electric field. Free radicals are paramagnetic because they contain unpaired electrons that have magnetic moments that are attracted to the magnetic field. In case of singlet state, when all electron spins are paired in the molecular electronic state and electronic energy levels do not split when the molecule is exposed to the UV radiation. So if there is an n number of unpaired electrons, it means that n plus 1 fold degeneracy, equal energy state, will be associated with the electron spin regardless of the molecular orbital occupied. Thus, if no unpaired electrons are present, n equal to 0. So according to the formula, n plus 1 spin state. So if you can see for the doublet state, a doublet state occurs where there is an unpaired electron that gives two possible orientations when exposed to UV radiation and imparts different energy to the system. Now, singlet or a triplet can form where one electron is excited to a higher energy level. In an excited singlet state, the electron is promoted in the same spin orientation as it was in the ground state. In a triplet excited state, the electron that is promoted has the same spin orientation to the other unpaired electron. Okay, so once a molecule has absorbed energy in the form of electromagnetic radiation, there are a number of routes by which it can return to the ground state. If the photon emission occurs between states of the same spin state, it is called fluorescence. If the spin state of the initial and the final energy levels are different, the emission is called phosphorescence, right? So, in the diagram, this is depicted by a longer wavelength lower energy and a shorter length blue line. Since fluorescence is statistically much more likely than phosphorescence for most molecules, the lifetimes of fluorescent states are very short and phosphorescence somewhat longer. 3 non-radiative deactivation processes are also significant here: internal conversion or IC, intersystem crossing or ISC, or vibrational relaxation. It is an intermolecular process by which a molecule passes to a lower energy electronic state without emission of light, overlap of vibrational energy levels into electronic energy levels. So external conversion is basically a process in which excited molecules lose their energy due to collisions with other molecules or by transfer of their energy to solvent or other unexcited molecules. Therefore, the external conversion is influenced by temperature, solvent viscosity as well as solvent composition. Intersystem crossing is in this process spin of an excited electron is reversed and change in multiplicity results. Most common when vibrational manifold of overlap exists and when the molecule has a heavy atom constituent like bromine or iodine. Now, what are the factors that affect fluorescence? The nature of the chemical structure of a molecule in terms of flexibility and rigidity is of major influence on the fluorescence and phosphorescence signal. Molecules that have a high degree of flexibility will tend to decrease fluorescence due to high energy, higher collisional probability. However, more rigid structures have a lower probability of collisions and thus have more fluorescence potential. For example, biphenyl has very low fluorescence quantum efficiency due to the flexible nature of the molecule, while fluorine has high fluorescence quantum efficiency due to its rigidity. Now, the polarity of solvent, the polar solvent is preferred as the energy required for the P to P star is lowered. Highly viscous solvent is preferred since collisional deactivation will be lower at higher viscosities. And if solvents contain heavy atoms, fluorescence quantum efficiency will decrease and phosphorescence will increase. Effect of substitution. Substitution structure can also affect the fluorescence. Molecule experiences larger collisional deactivation at higher temperatures due to an increase in the movement and the velocity of molecules. Therefore, lower temperatures are preferred for analysis. Dissolved oxygen affects fluorescence at higher, larger scales. Molecules experience intersystem crossing due to its paramagnetic nature. Effect of concentration. The fluorescence is directly proportional to the amount of absorbed radiation. So, when the concentration of the fluorescent molecules increases in a sample solution, the fluorescence intensity is reduced. Now, if we talk about quenching, it refers to any process that decreases the fluorescence intensity of a sample. So, a variety of molecular interactions can result in quenching, like molecular rearrangement, static quenching, and collisional quenching. Now, what is excited state reactions quenching? Such reactions occur because light absorption frequently changes the electron distribution within a fluorophore, so which in turn changes its chemical or physical properties. For example, a neutral solution of phenol red can lose the phenolic proton in the excited state. It involves the migration of a group or an atom from one center to another due to light and heat within the same molecule. For example, lumine santonin, a hydroproduct of santonin obtained by molecular rearrangement. Collisional quenching is basically when the excited fluorophore experiences contact with an atom or molecule that can facilitate non-radiative transitions to the ground state. Common quenchers include oxygen, iodine, cesium, acrylamide. For example, quenching of quinine drug by chloride, iron quenching by tryptophan. Static quenching occurs at the ground state of the fluorescent molecule. It can be simplified by the following mechanism. Concentration obviously is a kind of self-quenching. It occurs when the concentration of the fluorescent molecule increases in a sample solution. The fluorescent intensity is reduced in a highly concentrated solution. Chemical quenching is due to various factors such as change in pH, presence of oxygen, halides, and electron withdrawing groups, heavy metals, etc. Change in pH, like aniline at pH 5 to 13 gives fluorescence when excited at 290 nanometer, but pH below 5 or pH greater than 13 does not show any fluorescence. Oxygen leads to oxidation, and halides like chloride ions can lead to quenching. Heavy metals presence can also lead to quenching because of complex formation and collision. I have talked about instrumentation. I am not going into again detail. We have talked about what fluorescence is, Jablonski diagram. We have talked about Stokes shift. We have talked about so absorption is an instantaneous event. It occurs so fast that there is no time for molecular motion during the absorption process. Thus, absorption spectra are not sensitive to molecular dynamics and can provide information on average solvent shell adjacent to the chromophore. In contrast to absorption, emission occurs over a longer period of time. So the length of time fluorescent molecules remain in an excited state provides an opportunity for interactions with other molecules in solution like oxygen. Other examples of dynamic processes in solution involve fluorophore solvent interactions and additional diffusion. So quantum delay is basically the number of photons emitted by the number of photons absorbed. So in a given solvent, the quantum yield of a particular fluorophore will be fixed. Because of this, every fluorophore will have a characteristic fluorescence spectrum. So energy may be lost in vibrational transition, collision with the solvent, heat transfer, etc. Only a part of the light absorbed is emitted. It's because of this that quantum yield in most practical cases is not equal to 1. So quenching of fluorescence may also occur due to the presence of some foreign molecule in solution which is acting as a quencher, or due to some structural rearrangement in the solution, like protein, which drives the fluorophore to a conformation where it is in the vicinity of a quencher, like any amino acid residual or disulfide bond. So fluorescence intensity may also decrease with the transfer of the emitted energy to some other chromophore, which absorbs it at energy. So this phenomenon is called FRET. However, FRET and quenching should not be treated synonymously. So I've talked about some light sources and all of that, detectors. We will not go into too much details. Applications also we have talked. So biological functions, there's a lot of biological functions. I'll upload the slides, maybe you can take a look. Now, lifetime measurements can reveal how each fluorophore is affected by interaction if there are more than one fluorophore. So, it basically distinguishes between static and dynamic quenching. So, resonance energy transfer can also be best studied using lifetime. So, fluorescence lifetimes are typically independent of probe concentration, hence often used for cellular imaging. So fluorescence lifetimes tend to be shorter in more porous environment because larger dipole moments of surrounding molecules can increase the efficiency of energy transfer. So it is useful in elucidating changes in the microenvironment like viscosity, pH, polarity, solvation, size and shape of molecules, molecular interactions, inter and intramolecular distances, kinetic and dynamic rates, dissolution of molecular mixtures. All of these. So molecules are excited by a very short pulse close to lambda pulse, not lambda, delta pulse at T0, and the decay of the fluorescence intensity is measured usually by time correlated single photon counting. So excitation state is harmonically modulated with circular frequency omega, and so is the emission. So the fluorescence lifetime can be deduced from the phase shift phi and the modulation m. Not going into too much detail. We have talked about fluorescence and all of that, phosphorescence. We talked about the different applications of fluorescence. There are various fluorescence antibody tests for immunofluorescence we use. Basically, the principle of fluorescence. In flow cytometry also, we basically cells are sample labeled with the appropriate fluorescent antibodies, and cells in suspension are passed through machine in a single file in a stream of fluid. Stream is focused through one or more laser beams measuring light scatter and fluorescent characteristics. Fluorescence detected by photomultiplier tubes. So, not going into details. So, which of the following is not a common application? It will be 2. So, I'll wait for a few minutes. If anybody has any queries, you can ask now. Then we'll move on to the next question for today. Okay, so moving on to the next question for today. Sorry. Okay, I should have just give me a minute. Really sorry. They should come with animation and not immediately. So yeah. So the first step in a two-dimensional gel electrophoresis generates a series of protein bands by isoelectric focusing. In the second step, a strip of this gel is turned 90 degrees, placed on another gel containing STS, and an electric current is again applied. In the second step, option A, the individual bands become visualized by interacting with protein-specific antibodies in the second gel. Option B, proteins with similar isoelectric points become further separated according to their molecular weights. Option C, the individual bands become stained so that the isoelectric focus pattern can be visualized. Option D, the proteins in the bands separate more completely because the second electric current is in the opposite polarity to the first current. I'll give you guys a few minutes to come up with the answer, then we'll discuss. Anybody? 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 Okay. So the thing is, I'll discuss the answer. So the right answer will be proteins with similar isoelectric points become further separated according to their molecular weight. So two-dimensional gel electrophoresis is basically a powerful technique used to separate complex protein mixtures with high resolution. It combines two independent separation principles. Step 1 will be isoelectric focusing, that is separation by charge. Proteins are separated in a pH gradient, immobilized pH gradient strip. Each protein migrates until it reaches the point where its net charge is 0. Its isoelectric point, that is pi. The result is a series of protein bands or spots distributed along the pH gradient. At pi, protein has no net charge, that is the migration stops. Step 2 is STS phase, separation by molecular weight. The IEF strip from step 1 is equilibrated with STS, which denatures proteins and gives them a uniform negative charge. The strip is then placed perpendicular 90 degrees on top of an STS gel, paid gel. When electric current is applied again, proteins migrate according to their size. Smaller proteins move faster through the gel. So therefore, in the second dimension, proteins that have similar isoelectric points in the first dimension are separated according to their molecular weight by STS page. Okay. So the first option that is written here, the individual bands become visualized by interacting with protein-specific antibodies in the second gel. That is basically what happens in Western blot, not in 2D electrophoresis. Second option is correct, as we discussed. The third option, the individual bands become stained, that is staining or Kumasi silver happens after electrophoresis is complete, not during the second step. On opposite polarity current, the polarity is not reversed. The separation mechanism simply changes from charge based to size based. Okay. So we'll not go into DNA or agarose gel. We'll directly jump into. We have already talked about electrophoresis in our previous classes. So we'll only talk about page. Page can be either native page, like protein is prepared with little disturbance to the cellular material. Proteins are associated. Movement of samples through the gel can be inconsistent. Or STS page. Sodium rhodecyl sulfate is a detergent. Protein coated with a negative charge in proportion to its molecular weight denatures and unfolds protein. Reducing agents break amino acid crosslinks. The polyacrylamide gel basically creates tunnels in gel for molecules to move through. So before STS, after STS. We have discussed in yesterday's class. It separates proteins from each other, like proteins separated by size, isoelectric point, and it determines molecular size of protein, quantifies the amount present, it displays impurities. Used in Western blot accessed by antigen interactions. Okay, so determines molecular size of protein, quantifies the amount present, displays impurities, used in Western blot assays by antigen interactions. Now, for determining molecular weights, we need to run standard molecular weight markers on the gel, right? Run unknown protein on the same gel. 1 second. Okay. And we can run unknown protein on the same gel and create a graph of the molecular weight versus distance molecule has moved. Using the distance unknown has moved, determine the molecular weight from the graph. That is how it's done. We will not go into Western blot. We have talked about STS in the previous section. We will directly go to isoelectric focusing. So what happens? Separates proteins by charge, separates amphoteric molecules in a pH gradient. Amphoteric molecules and molecules whose charge is dependent on pH, they all have a pi point where they have neutral charge. When a pH gradient is created across a gel, the amphoteric molecules will be pulled by the electrode until it reaches its pi point. Once it is neutral, it is no longer affected by the electrodes. Okay. So pK of the carboxylic acid is around 2.2 and pK of the amino group is around 9.4. So when pH is greater than pK, the group is deprotonated. When pH is lesser than pK, the group is protonated. So regarding two-dimensional electrophoresis, it separates by both size and charge. So IAF or isoelectric focusing is first used on a gel strip. The strip is then mounted on a gel slab and SDS page is used. Proteins can be later removed from gel to identify basically. The proteins great for large-scale comparisons like proteomics. What is capillary electrophoresis? Basically, alternative method to gel electrophoresis. Reagents are dragged through a capillary tube rather than a gel, and detector uses UV light absorbance readings to identify whether a protein separation has passed through. So we'll not go into these details. So this is how it's done. First, separate in first dimension by charge according to electrophorecing. Protein mixture is separate given from here, so it will stop at the position where its PI or isoelectric point is. Then this will be kept like this in a different direction, vertically and horizontally and then it will in second dimension it will be separated by means of weight. It looks like this, something like this. Okay, so separation of proteins by the PI value, soaking the gel in SDS solution and fitting it on an SDS PA gel. For acrylamide, separating the proteins by molecular mass with SDS page. So that is the decreasing molecular mass. So this is how it's done. So conventional electrophoresis techniques can separate up to 100 different proteins on one run. Typically, cell or tissue extracts contain thousands of proteins, most of which will not be resolved into single bands using a separation based on any one parameter such as size or net charge. For any one size range, there is a high probability of more than one protein out of thousands falling into this range. The separation on the basis of two parameters, usually size and isoelectric point, lowers the probability that two proteins will overlap and allows the resolution of thousands of protein species in one gel. So it's basically introduced by O'Farrell and Close in 1975. Isoelectric point 2D page provides the highest resolution separation method for proteins. Following a first dimensional IEF, proteins is subjected to SDS page in a perpendicular direction, and the technique is a true orthogonal procedure in that the two separation mechanisms are based on different physical principles and the two separations are done at right angles to one another, geometrically orthogonal. First is protein sample preparation, first dimensional electrophoresis, second dimensional electrophoresis, gel staining and protein visualization. Gel imaging, protein analysis, protein identification. So it's also known as electrofocusing. It's a technique separating the charged molecules, usually proteins or peptides, on the basis of the isoelectric point. So it employs a pH gradient extending the length of an electrophoresis gel. A protein stops migrating. A protein stops migrating when it enters the zone in which the surrounding pH equals the isoelectric point. At any point other than any other point in the gradient, the protein acquires a charge which causes it to migrate towards its PI. So basically, a commercially available immobilized pH gradient strip is used. IPG strip consists of an acrylamide gel that contains pores to prevent a sieving effect based on protein mass with the pH gradient, and various gradients are available. So for wider range, for example, 3 to 10 are used for whole proteome analysis, and lower ranges are used for more specific applications like pH 5 to 8. So the pH gradient in the IF gels is generated by the inclusion of ampholites, low molecular weight amphoteric molecules. A mixture of ampholites is used, each having a different PI. So like protein molecules, the ampholites migrate through the gel until they reach a region where the pH is equal to their PI. Unlike the proteins, ampholites are present in high enough concentration to change their local pH. So the gel is set up with a uniform mixture of ampholites throughout, and its anodic and cathodic ends are immersed in dilute acid and base respectively. So ampholites near the ends of the gels will be positively charged near the positive electrode and negatively charged near the negative electrode. They therefore begin to migrate into the gel, with the most charged, that is the ones furthest from their PI, moving the fastest. So over time they separate into zones of defined pH. If the ampholite system is well designed, a smooth gradient of pH is created with no abrupt changes or steps. So various mixtures of amphoteric substances have been used as amphorites, amino acids, proteins, and polyacidic, polybasic synthetic molecules. Natural amino acids have poor conductivity and poor buffering capacity in the zwitterionic state. making them poor candidates. Proteins can be good ampholytes, but they interfere with analysis of the sample by introducing new proteins into the mixture. So polycarboxylic acid polyamides are the most commonly used ampholytes, have excellent buffering capacity and conductivity across a broad range of pH, usually provided in the molecular weight range of 300 to 500, which is small enough to avoid interference and with the most subsequent processing. The sort of disadvantage is that they may bind tightly to the proteins due to ionic interactions and can be very difficult to remove. So sample is usually combined with kaolin ampholytes to assist in migration. Ampholytes are a mixture of charged molecules with a range of pIs that matches the pi range of the IPG strip. The migration of the ampholytes encourages the sample molecules to move along the pH gradient. Commercial ampholyte mixtures of variety of pi ranges are available. After separation across the pH gradient, the sample is further separated in 2D pH or analyzed in the case of fractionation or mass spec. So the application can be to study the entire protein of an organelle or bacterium to identify and determine differential abundance of proteins for various downstream applications like biomarker discovery and all of that. To examine post-translational modifications of proteins like phosphorylation, methylation, glycosylation, acetylation, etc. and fractionation of proteins or peptides prior to mass spectrophotometry. So you can see how it's done basically, 7 different types of separation. So I am not going to detail, I have already discussed. We've already talked about SDS page. Isoelectric focusing also we have discussed. You can see an isoelectric ampholyte solution is incorporated into a gel. Stable pH gradient is established in gel after the application of an electric field. Protein solution is added and the electric field is reapplied. So after staining, proteins are shown to be distributed along pH gradient according to their pi values. So this is achieved by the ampholyte and must have falling prop, like must dictate pH course, should have conductance at their pi, low molecular weight, soluble in water, low light absorbance and all of that. We have talked about two-dimensional. This is how it happens. Not going into too much details. Coming back to the question that we actually started our, this is how 2D occurs. First dimension isoelectric focusing and second dimension is SDS page. You just need to remember that. So coming back to the question that we started with basically. So the first step is basically 2D isoelectric focusing, second step is SDS page. The proteins with similar isoelectric bonds become further separated according to the molecular weights. So any questions guys? I'll wait for 1-2 minutes. If you have any questions you can let me know. Then we'll stop the session. So if you have no more questions, we can stop the session for today. Thank you so much. This was the last session. Really glad to have conducted these live sessions. Thank you so much for being there. I'll upload all this recordings on YouTube and all the slides, so you can come back to this whenever required. Thank you.